An antigen, AG, or antibody generator, is any substance that elicits an immune response in a host animal, such as a mouse. Foreign substances, including proteins, can act as an antigen. One can use a host animal to produce antibodies that recognize a protein of interest, protein X. The first step in preparing antibodies specific to protein X is to inject a mouse with a solution of the purified protein. Usually an adjuvant is added to the purified protein to boost the immune response. The size of the protein antigen can also affect the quality and quantity of the antibody produced. Small peptides are usually non-antigenic and need to be cross-linked or conjugated to another antigen to elicit an immune response. Once protein X is injected, it interacts with B lymphocytes to produce a pool of IgG-class antibodies against the protein. These are polyclonal antibodies that recognize multiple sites on the protein antigen. Since mice are small, the amount of polyclonal antibodies that can be isolated from sera is usually insufficient for research purposes. Polyclonal antibodies are usually produced in larger mammals. However, mice are commonly used to produce monoclonal antibodies, which recognize only a single site on the antigen. Each B lymphocyte can produce a single type of antibody specific for a single molecular determinant. Within the spleen, the B lymphocytes expressing antibodies specific to determinants of protein X will bind the protein. This triggers a series of events that stimulates a cell to grow and divide, resulting in a colony of cells. Usually a single exposure of a mouse to an antigen is not sufficient to generate a strong immunogenic response. It is typical to immunize the mouse a total of 3 times, at days 0, 14, and 21. This increases B lymphocyte colony formation and the antibody titer. Next, a population of spleen cells is removed from the mouse. This includes the B lymphocytes with protein X-specific antibodies and nonspecific lymphocytes. The spleen cells die after only a few cell divisions. To allow the spleen cells to grow indefinitely, they're mixed with HGPRT- mouse myeloma cells. Myeloma cells are immortal and will grow indefinitely in culture. They can also fuse with other cell types, creating hybridomas that are also immortal. HGPRT- myeloma cells are used for the hybridoma fusion. They have a mutation in the enzyme hypoxanthine guanine phosphoribosyl transferase, HGPRT, required for a secondary rescue pathway for the production of purine nucleotides. When the cells are grown in a special medium, called HAT, that requires the use of this pathway, the HGPRT- myeloma cells cannot survive. The hybridoma cells are the only cells that can survive in the HAT-selective medium because they have the wild-type HGPRT enzyme from the spleen cell and the immortality from the myeloma cell. The remaining hybridoma cells are diluted and plated so that only a single cell is in each well of the culture plate. The cells are then allowed to grow, forming a colony of identical hybridoma cells that will produce a single type of IgG antibody. The hybridoma clones are tested for production of antibodies specific to protein X. The hybridomas can now be used to produce pure monoclonal antibodies for use in cellular and biochemical experiments. Since the established cell line is immortalized, the hybridoma can be preserved and used to produce the same monoclonal antibody for many years.